Hello everybody and welcome back. This is part 2, 11.1, antibody production and vaccination. In this video lecture we're looking at the specific immune response. Now the actual system itself is often called the challenge and response principle, with the challenge being the challenge presented by the pathogens in the form of the antigen, and the response being what the body does to combat that the invading pathogen itself. So generally the first stage of this is what we can see here at stage 1, is where macrophages will ingest pathogens and display the antigens from it. So the antigen, here's our pathogen here being ingested, okay, and the antigen is being pushed out of the membrane of our macrophage, okay, or it's being presented, we can say it's being presented. Now macrophages, it's important to realize that macrophages are non-specific. Okay, they are not specific to any particular antigen. They do have surface cell receptors, so they can notice that they can recognize things that are foreign to the body, and they obviously recognize cells that belong to the body itself and do not engulf those. Okay, but they're not specific in their response. Their only response is to engulf the pathogen itself and present the antigen through its membrane. The next stage we see is a helper T cells. These are specific to the antigen, and what they do, they have a receptor on their membrane which is specific to this antigen. So not all helper T cells will have this specific receptor here. Okay, some helper T cells will have other receptors, and of course they won't be able to bind, but this one does have the right receptor, and it's able to bind to it. So once they're bound together, the helper T cell is activated by the macrophage. The activated helper T cell then binds to lymphocytes, okay, called B cells, which is happening down here at stage 3. Okay, only B cells that have the receptor protein to which the antigen binds have a receptor protein and will undergo the binding process that we can see going on here. So again, it's not all B cells which will undergo this process. The helper T cell activates the selected B cells both by means of the binding, so this is the binding here, okay, and by the release of a signaling protein. So you can see these signaling proteins being released, which are going to activate this B cell. So what generally happens next is our mature B cells, or B cells, are going to mature and grow into plasma cells. And these plasma cells are very important because these are our main antibody producing cells. So what we're going to do here, I'm going to come back to this slide in a second, but we're going to fast forward to the next slide and show you what an activated B cell looks like. So this is a B cell. What we can see here is a cell membrane. Around here we can see mitochondria, we can see a large nucleus inside here, and then we can see all these areas inside here with these black dots surrounding them. Now what this is inside here, it's a very extensive network of rough endoplasmic reticulum. And what that rough endoplasmic reticulum does is it manufactures, modifies, and transports proteins, which is in this case is the specific antibody that is being produced. Now the cell produces a lot of the same type of protein, meaning that only a small range of genes is expressed, which is lower than the typical cell. Okay, and that kind of is seen here. You can see inside the nucleus here that it's quite dark. Now this dark DNA, or this dark area inside here, is dark DNA, and this kind of puts the idea forward that this is DNA that's very, very rarely, if ever, actually being expressed. So the genes within that DNA is not being expressed. So this is quite a common feature of these B cells, is that a lot of the DNA actually isn't being used to express genes. Okay. So let's pop back now. So here's our activated B cell. And this is going to divide many times by mitosis, generating a clone of what we call plasma cells, which is what we just looked at. Now the generation of these large numbers of cells that can produce only one specific antibody is known as, we call that clonal selection. Okay. Now those antibodies, of course, that are produced by these plasma cells are secreted, and these will destroy, hopefully in many different ways, which we'll look at a bit later, which will help in destroying our antigen or our pathogen that the antigen belongs to. Now, one thing we haven't discussed is memory cells. So we said that most of our mature B cells become plasma cells, okay, which is here, but also a very small number of those B cells will become what we call memory cells. So what these memory cells do is they will remain inactive, okay, unless the same pathogen infects the body again. If this happens, they will become active again and respond very rapidly, okay, producing antibodies very quickly. Immunity to an infectious disease involves either having antibodies, okay, so having antibodies circulating within the bloodstream, or having the memory cells that allow for the rapid production of those antibodies. Okay, so that when we look at the immunity shortly, you'll see that what happens within that secondary infection. But before we do that, we're going to look at the roles of antibodies and what they do. Okay, so we're going to start here with, you can't see it very clearly because my image has been blocked out by the mouse cursor. It's called opsonization. Now what this means is that you can see here we've got a phagocyte, you've got these Y-shaped antibodies, and you've got the pathogen in the middle here. So what happens there is the antibodies are causing the pathogen to become more recognizable to the phagocytes and basically mean they're more readily engulfed. Once bound, they can link the pathogen to phagocytosis, and so they're all being engulfed within the phagocyte. Another way that can help is through what we call neutralization. So there's two types of neutralization. Here you can see that the antibodies have surrounded the virus or a bacterium, and they're preventing them from docking with the cells of the host here. Okay, so they're stopping them from docking. Okay, they can also neutralize toxins. Okay, what that basically means is that they can bind to the toxin-producing sites on the pathogen, okay, and stop these toxins from being released into cells in the body that are susceptible to these different types of toxins. We've got what we call agglutination. Agglutination is what we discussed earlier with what happens to red blood cells. If you have a blood transfusion and you receive the wrong type of blood, it's the specific immune response. Basically, the antibodies will cause those cells to stick together and clump together. Now that obviously is that large agglutinated mass is often filtered by the lymphatic system and then engulfed. They're often then engulfed by phagocytes. Can be, of course, very dangerous if it happens in your blood supply, and they agglutinate because it can cause blockages in your blood. The last one here is what we call the activation of complement. Now in this one, we have a complement system, and it's a collection of proteins. So you've got your antibodies, remember, which are proteins, and you've got a complement or a complementary protein here. And basically these guys work together and they cause the perforation of the membrane. Okay, so they're going to cause the membrane to burst open slightly. What generally happens then is water and ions are able to enter the cell, okay, filling it up and cause it to lyse, obviously, which means to sort of burst open. Okay. Now, of course, you know that we can become immune to diseases. We become immune, we gained immunity to lots and lots of diseases over the course of our lives. Now, that immunity is usually due to either the presence of the antibodies that recognize antigens associated with the disease, or to memory cells that allow for the production of those antibodies, just like we discussed earlier. So immunity develops when the immune system is challenged by the specific antigen and produces antibodies and memory cells in response. Here you can see the primary response look. And in that primary response, obviously it takes time, okay, and the concentration of the antibody as it gets to its highest number, okay, this is taken up to about 4 to 5 days before producing the maximum number of antibodies, okay, and you're becoming less symptomatic, okay. After the infection, you've got rid of the infection, the number of antibodies you can see is staying in the blood, but it's decreasing, all right, and it's decreasing quite quickly, okay, not as quickly as they're produced, but still it's decreasing quite quickly, okay, almost down to 0, but not quite. There's always a few in there. Now, if there's a secondary encounter, you can see here that response is faster, okay, with a huge number of those antibodies being produced, okay, within this period of time here. And of course, we know that that's because we have the memory cells in there, which can very, very rapidly reproduce the antibodies that we need to combat that infection. Okay, so they're already there. The last thing we're going to speak about today is vaccines. Now, the vaccine is usually introduced into the body by injection, and what it usually consists of in that vaccine is an attenuated simply means weakened, but it's just a fancy word for weakened version of the pathogen. It also may be if it's not the actual pathogen itself or an attenuated pathogen, it may be a polysaccharide from the surface membrane which acts as an antigen. Okay, and basically what that the vaccine does is it triggers that primary immune response, okay, that we've just discussed in detail. And of course, during that primary response, memory cells are produced, which can then reproduce very, very quickly should there be a secondary infection. Now, what you can see here in this image, it shows a phagocyte, which is this guy here, okay, engulfing Mycobacterium bovis. Now, this is the strain of bacterium used in the vaccination for TB or tuberculosis. These bacterium are attenuated, which means they've been weakened, so they're not very, very strong. And they're nowhere near as pathogenic as the TB itself. The vaccine primes the immune system to produce antibodies that act on both species of bacteria. Okay, and of course we're producing those memory cells which can reproduce those antibodies very quickly. But of course the disease is not caused by Mycobacterium bovis. Okay, so you don't become symptomatic of TB. Now, obviously, if there is now an infection with TB, because the antigen is the same as Mycobacterium bovis, okay, we've got a very, very rapid secondary response to that. Anyway, that's all we're going to discuss today. I know it's been slightly longer than you probably thought it would be. Thank you for listening.